So I begin today by acknowledging the traditional custodians of the land which we meet on and pay my respects to the elders past and present. I extend that respect to Aboriginal and Torres Strait Islanders peoples here today. Hello everyone. My name is John. Thank you for joining us at the 2024 NCARD Public Lecture, whether you're here in person or attending online. We at the National Centre for Asbestos-Related Diseases, or NCARD, are delighted to welcome you. The theme for this year's seminar is Asbestos, New Frontiers in Management, Science and Care. Every year, over 4,000 Australians die from asbestos-related diseases. That's the equivalent of one person every 2 hours. Of these deaths, more than 700 are due to mesothelioma. Western Australia continues to have one of the highest incidences of mesothelioma in the world. Despite asbestos being banned, it is ever present in our surroundings, and the risk of exposure is ever present. NCARD is the largest research centre focused on mesothelioma research. Our research team is dedicated and committed to understanding asbestos-related diseases and translating our findings to better patient treatment and care. We have been and continue to be leaders in research excellence, driving international collaborations with scientists and clinicians both within Australia and around the world. Supported by government funding, public donations, and disease foundations, our work has produced groundbreaking research that has improved the lifespan and quality of life for mesothelioma patients. In particular, we'd like to thank the Willoughby Foundation for their generous and ongoing support of NCARD research. We also like to thank Tom Rickaby for his extensive fundraising efforts by doing the Cape to Cape walk. Thank you, Willoughby Foundation. Thank you, Tom. With these funds, our goal is to continue to advance treatments and outcomes for those affected by asbestos-related diseases. Tonight, we are fortunate to have 3 experts to share their latest insights and findings with us. Following their presentations, there will be opportunities for questions and discussion. We also invite you to join us for refreshments after the seminar. In the foyer, you have seen there are posters prepared by our research team, including work for our students. These students represent the best and the brightest and the future of mesothelioma research. We encourage you to go through the posters, ask them lots of questions, and for the project and the poster that you like the most, we'll be having a popular vote prize after. So if you can vote for your favorite or what you think, basically your favorite poster, that will be very helpful for us. So your input is greatly appreciated. We hope tonight will be an opportunity to continue our conversation with all of you, patients, carers, advocates, and others who have been affected by asbestos-related diseases. Okay, so it's my pleasure to invite our first speaker, Mr. Samuel Jackson. Samuel Jackson is the Projects Director at Thuroona Services and HAZRAD Australia. He holds the asbestos license for Thuroona, is a licensed asbestos assessor, and serves as the board member of the Faculty of Asbestos Management of Australia and New Zealand. He actively participates in several local and national asbestos special interest groups and maintains a strong network around the state, particularly emergency response. Samuel has been at the forefront of many emergency response efforts to natural disasters across the state, providing timely advice and rapid response services. Notable examples include his work at the Ruraloo and Gidgegannup bushfires, Cyclone Seroja, the Bridgetown and Hester fires, and the recent Bunbury tornadoes. So without further ado, it's our pleasure and honor to have Samuel up to share his presentation with us. Samuel, please. Is the spotlight on?
Hello, everyone. As mentioned before, my background is predominantly asbestos removal, environmental site investigation for a start. One of the other things that we focus on is not just emergency response for asbestos-related issues. We actually undertake controlled waste, dangerous goods, explosives, radioactive work across Western Australia. So some of the topics I'm going to present here, we only had a really short amount of time, so if you want to see a few things that are a bit different, I'm happy to chat at another time. So the first part on this is, I'm sure everyone in the room knows a little about Whitnum. Whitnum was actually, the town site was demolished just finishing on just a year ago now. And we undertook the site demolition of 37 odd structures and actually buried it within the town site itself. And I'll go through a little bit of the process and why we did particular things to give you a little bit of a different perspective on risks that are currently here now. So they're not just in the past. So quick photo there. Those familiar, that's Dock Holidays, which is at the beginning of the town site. Pretty well known location. That was the last building to be demolished within the town. I'll do a quick brief on asbestos and where it sits with Whitnum. Asbestos is a generic form of the fibrous silicate minerals. Essentially, when it crushes and forms into long fibres instead of crushing into dust like normal rocks. Whitnum itself, it's crucidolite, so it's a blue asbestos. You'll see some of the photos. It actually is very blue, but it's also grey. So when you look at it and it's wet, it's predominantly blue, but when it's dry, it's actually a bit of a grey colour. For some perspective in the photograph here, asbestos compared to some of the other material, look, you've got sand up at the one end of the spectrum and you've got asbestos particles, which are quite far down. During all our work, whether it's emergency response and actually doing plan removal works, sometimes it's very hard to explain to someone that you're looking at a bit of rock or a beach sand. The asbestos, even though you can see it, the things that you can't see are significantly worse. The location of Whitnum, so some may know it actually only takes a day and a half to drive from Perth. So essentially we can drive, leave at 4 o'clock in the morning and we'll get there the next day early in the morning, so it doesn't actually take too long to drive. Where is Whitnum from Perth? As I said, 15 hours, 14 minutes. It's not very far. Port Hedland, you're looking at about 3.5 hours. From Newman, it's about just under 3 hours. So the size of Whitnum, you've got something called the asbestos management area. Asbestos management area is just under 500 square kilometres. Now, I'm not sure if anyone saw in the media this afternoon, but there was an article published in the Business News where they're looking at potentially including the Millstream National Park in that area as well. Now, for those that don't know, Millstream is about 160 kilometres from Whitnum, heading north. We have worked on the Robo and Whitnum Road for a number of years for both main roads, Department of Lands and Heritage. And there are multiple containment cells, talking 200 kilometres away from Whitnum, buried underneath the Karratha Tom Price Road. So it's now called the Mandurana Red Dog Highway. Under that bitumen, there's 7 containment cells with material on that road, 160 kilometres from the actual town site. So this material isn't just located in Whitnum. Bit of a timeline of Whitnum. Essentially, 2023 is when we finished demolishing the remaining structures. A couple of quick photos. You might have seen some of these before, but I'll just put them into perspective. This is one of the former, the mill and the mine itself. You can actually go where the just this spot over here is actually open to the public still. So every time we go up there, we re-weld the metal grids on, and every time we go back, someone's used the angle grinder to climb up the tailings to get inside the mine shaft, and that mine shaft goes about 6 levels underground. So we're up there very frequently. This is some of the workers in the mill. Photo on the left, pretty familiar as well. They have a competition on how quickly they can load drums full of asbestos tailings. Obviously, massively high exposure there. On the right-hand side is one of the trucks. Love to show you a bit more of some of the other projects we've done relating to these cleanup of these trucks. But essentially, this would drive from the town to Point Sampson along that road on Whitnum Road, dropping bags as they go, truck rollovers, and over time the grader goes on the road and just grades it into the road, mixing it with the dirt. The Whitnum Golf Course, just outside of town. The Pictures Theatres there. I've actually got one of the chairs of the Picture Theatre back in our warehouse when we did the demolition work. The Fortescue Hotel. I'll quickly jump over these because I'm really limited on time, but there's quite a significant amount of people that went through Gairdner with a lot of related deaths, unfortunately. Quickly onto the site itself. So this is the town site of Gairdner, or the former town site of Gairdner. In the boxes here is where we actually built the containment cells. So essentially it was deemed unsafe to remove the material from the town site. So we actually built a containment cell, excavated it from the ground and demolished the buildings, bringing them straight to the containment cell. This road here goes to the Oski Roadhouse, which is about a 49-minute drive. The Great Northern Highway is off to the right-hand side. So one of the issues we had was members of the public coming into our work area unprotected. So obviously when we're undertaking work, there's a significant level of risk related to doing that actual work. So essentially we created a 1.2 kilometre water barrier to block the actual town site off itself along the edge of it. And we had people bush bashing through with four-wheel drives trying to get around the roadblocks to go into the tailings. Essentially, these are water bollards full of water with some of the personal protective equipment that our crew are using. So it's full face PAPR respirators. Not the most comfortable thing to wear in 40+ degrees, but that's the standard of work. If you're outside of the vehicle, you're wearing PPE. Road blockages. So this is an example of one of the road blockages that we built, approximately 4 metres tall. This is to stop, because they were driving around where we had roadblocks, we had to physically start putting in further measures. Before we actually started the work, we flew a drone from the opposite side, which there is something called the Carragini Eco Retreat up the top of the hill. So we flew some drones through to check if anyone was in the gorge. I drove in there myself and had to have a couple of conversations with people that were still in the town and in the actual gorges themselves. You'd be surprised how many people actually go in there and camp. This sign here is a corker. This is one of the multilingual signs that were put up by us back in 2018. During COVID, it was a pretty interesting time. You couldn't really stay at home, so where else would someone go but Gairdner? So this particular sign is about 7 kilometres from the town site into the actual gorge itself. And you had all sorts of people, zombies in this area and all sorts of interesting things. When I talk about people coming in, this is a pure example. This is a family with a couple of kids in a caravan that had driven from South Australia to come into the Gairdner town site to have a look at what was going on because they heard it was getting demolished. They actually drove from South Australia to try and get in, and they were obviously they're repairing a flat tyre on their caravan there, and they've come across the roadblock with me there. Had a conversation. This is a bit of a drone view from our compound. So we had a compound set up within the asbestos management control area, which you wouldn't normally have. So typical asbestos removals, particularly friable projects, you're on the edge of what's called the hot zone. So you're in a cold zone where you can wear no PPE versus having a controlled area inside the zone. It presented a whole heap of risk challenges that we had to get through, including when we're building the actual dome, coming across raw tailings that are sitting on the ground that we had to deal with before that. Vehicles, you might ask, how did we get around? Well, the vehicles are actually pressurised. So this particular bit of kit is about $17,000, and there was 8 bits of kit that had to get fitted to different vehicles. So water carts, trucks, excavators, light vehicles for supervisors. It's a positive pressure system that is HEPA filtered both in the cab and then it filters going out of the cab. One of the things people don't probably think about is you've got asbestos, but if you're in the cab for a long time, you've also got CO2. So there are standards around reducing CO2 build-up because of fatigue inside the cab. So you've got a primary risk, a secondary risk. Some may ask, how do you control dust? Well, water carts, one of the risks that we looked at was filling water carts So we actually got all the generators and water tanks are actually remote controlled. So you drive up to the water tank, hit the button, it fills itself. You would know when it's full because all the water's gushing out the back of it. We actually tapped into the former town site bore and got that operational. So the only water source in that area was either FMG's Solomon mine, which was about 50 minutes up the road, which we had to use in the early days of setting up before we got the bore operational, and then the town site itself. Some may ask about monitoring. There was a very extensive monitoring program on this, testing some new technologies. This particular yellow box is the Alert Pro, which is a real-time monitor that's been tested in industry. It basically gives you alerts instantly if particle load goes above a particular level. And you'll see the 3 low-flow pumps up here. One of them is respirable dust. We had standard membrane filter method. And we also had a secondary gold filter that was set up. We actually set up something a bit different. So an electron microscope was imported from the Netherlands, and the mobile laboratory was set up at the Oski Roadhouse. So essentially all the samples were shifted from town, collected, and then went back to the Oski, put through membrane filter method and through SEM to get real results daily. So it might be in the Pilbara, but there's no excuse not to have monitoring out in the field. It might cost a bit of money, but it is what it is. Some may not have seen the tailings itself. You've probably seen mountains of stuff. This is some air monitoring equipment we set up in the actual tailings to compare what was happening in the gorge versus what was happening in the actual town site itself. Dust control. So when I talked about filling of water carts before, we had the main source of dust was actually not the demolition work. The main source of dust was actual dust coming off the road. This road is the source road for trucks going to Solomon. You can't come through the other way because of the restrictions on width. So all the toll fuel tankers that come to put diesel in the Solomon mine, which I think there's 7 quads a day, so 4 trailers. I'd like to show you a photo of the dust, but I'd probably get toll coming to sue me or something. But essentially when the trucks are going down the road, there is a cloud of dust over a kilometre long. And that dust was coming into our work site, overloading our monitors. So it wasn't actually our dust. So we needed to control the dust. And we used a mixture of a fibre suppressant, which has got a surfactant in it to put in the water trucks to break that surface tension on the ground. Quite a challenge, that one. I mentioned before about PPE. You'll notice this gentleman over here. Some of those buildings were actually built not with normal sheets. So you had asbestos, you'd had like a gypsum middle, and then asbestos on the other side. So once you crack the buildings, you couldn't penetrate into the actual substrate of the building material. So we had to physically get someone out and spray sometimes to control that dust. Again, dust control, demolition. Essentially, demolish the building, load it straight into a moxie, goes straight into the ground. Anyone got a flat before? Well, there's no RMA out there. So this particular vehicle we considered to be contaminated the whole time because there was staff, it was like a runner that was going around. So essentially these particular guys would go onto site through a decontamination unit out into the field, and they might have to fix things on an excavator or pick a spike out of the ground that's not going to spike a hole in one of the loaders. But they got flats. I probably think we went through over 20 on that particular work front, and they're out in 40 plus degrees wearing full PPE. Now you might think the job's done when you demolish the slab. Now these are some photos I'm about to show that haven't really been seen before. So there's the slab itself. That's what's inside the slab. So back in the 80s, they had a stabilisation program through the Whitman town site where the former Shire essentially put about 300 mil of fill across the site, across the town site. As we know, a lot of the kids played with tailings in the gardens, etc. One of the methods when they resurfaced the road inside, they used tailings to resurface the road as well, by the way. They sort of lifted the ground level around the site But what the issue was is to build these up to start with, actually filled them full of tailings and then concreted outside. We didn't know this until we got into it. Now, some in the media might have noticed there were some news articles around this. Now, when I saw this particular article, I was a bit concerned about what was happening on my job site because I was running the site essentially. So demolition of the town site of Whitman starts. Government warns users to stay away. This is a particular photo from the ABC. Now, I work a lot with emergency response and understand how the media work. And you can get a photo from a particular angle that may not actually be reality. The reality is that it was actually water mist from the cannons that we had in the containment cell. So we were digging in the cell, water mist appeared to be mist. There's a photo of the cell. There's a photo of the mist that's actually going over the cell. So when they say there's dust coming off, there's actually not. It was water. Would have been nice for them to say there was actually water controlling what was going on. You can see the height there. That's a 35-ton excavator that sits about 8 metres tall. So I think the bottom of the trench was about 12.5 metres deep. What there was in the containment cell though, and luckily we had an electron microscope up there because we hit some white material which we were a bit concerned about, but it was actually chert, which is a silicon dioxide. We had results in about 2 hours to tell us that it was safe because we thought we might have hit something that we hope not to, which was naturally occurring material. Legacy contamination, when I talk about distances from the town site itself, this is what the fibre bundles actually look like in real life. That's only what you can see, imagine under a microscope. This is material from a recent project that we've done up around there. So essentially out at the airport sits just outside of town to the north. The airport was resurfaced again with tailings. The airport's quite long, it's a couple of kilometres long. And when you start sampling throughout the airport, you're seeing fibre bundles and naturally occurring material. When I mentioned the road before, this is the actual road itself. You see the circles here. There are fibre bundles all through the road. That road's still open today. On the right is a picture of the close-up of the first. This material here is from here. And again, open to the public. That is me in the picture. That is raw crucidolite. They are the bags that fell from the truck I showed earlier that went from the town site to port. Some of these bags were remitted in 2019. Again, sitting on the side of the road, and that is made literally with 100% raw processed crucidolite fibres. You can see the PPE and whatnot. My colleague Kevin White, who's sitting in the audience, recently was up again at in that area, looking through the creek beds and other locations. You'll see fibre bundles in the creeks and the rivers. Again, you see the PPE in the background. There is Kevin. So Kevin is my offsider in the office, very, very knowledgeable guy that ran the last recent project for some sampling up there. Again, fibre bundles and naturally occurring material within a very short distance. I won't go too much into the other mines in Australia, but there are other mines that produce material, with Whitman being the largest. Now, what has asbestos mining got in common in Australia? We do a lot of removal, whether it's bonded or friable work. According to ASEA and the Victorian Asbestos Eradication Authority, there's about 6.4 million tonnes of asbestos material still in our environment today. So that is a number from 2024, and it's predicted to remain until about 2080. These are government release numbers as well. Just to put some into perspective, we're in 2024 over here. The lifespan of a lot of this material is at end of life. Flat sheeting, electrical panels, communication pits, corrugated roof sheeting, brake pads, vinyl flooring. This is the data from the manufacturers on the life of these products. we are again 2024, and you'll see there that apart from electrical boards, which have got a Zelmanite in it, they're all end of life. So whatever's sitting in the environment now is way past the time that that was recommended. Now, a little bit on emergency response. Love to go into it further, but not enough time, so I'll be brief. We are the DFES emergency response contractor for hazmat in WA. Do a lot of emergency response work for government and for private agencies. These are a couple relating to asbestos specifically. For the World of Bushfires, Thuroona Services, which is us, obviously, we demolished all the asbestos properties that were related to the World of Bushfires. That was funded predominantly by the federal disaster funding category C. Now, to get there was a bit of a process. So essentially, the Department of Water and Environment Regulation, once the fire went through, there was a bit of a tussle on who was going to manage it, whether it was Department of Finance or DWER. So the pollution response team, now called the environmental response team recently, they took control and essentially engaged us to go do site assessments immediately. So if we're talking smoke still coming out of the ground, we're on the ground the next day after the fire started. Worked out a strategy on how to get into these properties. A lot of them didn't have street addresses. They got lot numbers and the properties, a kilometre inside. And essentially, we had the crews out. We used a mixture of PVA, bondcrete, and paint. The blue paint means asbestos in the industry. So essentially, we go out, identify the properties that contain ACM. They were encapsulated, so a product was sprayed on to glue that fibre material to the ground or to the actual panels. To get to this stage, though, each homeowner had to sign on authority to act, which means essentially they're giving the government control of coming out to their property and doing something, because you can't just go onto someone's land and do that. There was issues around that. There was also issues in an emergency response situation. Members of the public don't really tell the difference between government contractor and government. So essentially there we're representing the state. So we would turn up to do work in conjunction with the City of Swan. And essentially we would have landowners that have just lost their cattle or lost their properties, come through with a lot of, people deal with grief differently, and you're getting, people, you're coming on there to do something to stabilise hazardous materials, and you're getting abused in a sense, and having to retract our staff from sites to reduce that confrontation and then come back at a later time. So 37 odd properties were encapsulated over the space of 7 days to make them temporarily safe. More pictures there. And that project was done over a 2.5 month span with a lot of pressure, both us and also the contractor that did the non-asbestos buildings. Another quick example, I could show you heaps of photos in this one. We actually rolled one of our trucks inside the asbestos cell on the last week of this fire cleanup. Ironically, it rained and it lost the truck in the cell and had to decontaminate the truck to get it out. Emergency response HESTA. So this is down in Bridgetown. This particular one, a fire started on a Western Power pole. I believe it's already gone to court, so I can say that. But essentially that fire started and there was 8 asbestos impacted properties on that one. What's not mentioned here is there we go. There was actually copper chrome arsenic coming from the timber treaters. So essentially the fire went through, burnt the treated material, and we had to deal with 24 contaminated properties around a Hester town site. So not just asbestos, CCA as well. Cyclone Seroja, that happened at the same time as Wooroloo and on the COVID shutdown in WA. When we got to town, again, my colleague Kevin, he was actually the first to respond from our team. This is a photo of the street. So that is a mix of asbestos and gypsum on the road and on the pathways. This was typical through a lot of towns, through a lot of streets in that town site. This is the PCYC building that come down. This is all material that we had to clean up. On the right over here was the path of the cyclone. You can see sort of has gone through here. Again, covered with asbestos-containing material. This is the school, the Kalbarri District High School. It basically lifted off the roof of the adjacent property and smashed the school. I'd love to show you some photos at a different time, but it actually went through the air sideways, and you can see the sheets that actually impacted the school, and that was quite interesting. Not the perfect example of things, but this is one of the roofs that had come off. Again, if we don't plan for removal now, Mother Nature is going to take its course. And as you can see, this is not a problem that's going away anytime soon. This is a bit of a sad one, actually. This material on the right here is the side of the driveway, this pile here. That's all asbestos sheeting. This is the noughts and crosses that kids were playing in the driveway. So again, this is not Whitnern, this is Kalbarri. You know, this is only a year and a half ago. You know, with noughts and crosses with asbestos, unbelievable. Bunbury tornado, we all know about 4 months ago, the tornado went through a town of Withers in Bunbury. We responded to that and did the clean up on behalf of Department of Fire and Emergency Services. Our friend Roger Cook over here came down and visited us as well in the Incident Control Centre. So essentially, you know, that town site, similar to Kalbarri, was shut off by WA Police, and you weren't allowed to get into that town site where the work was being done. Some of the examples are again similar to Kalbarri. A tornado forms over land, a cyclone forms over water. I didn't know that until the tornado hit Bunbury. I was wondering why they called it a tornado and not a cyclone. It actually formed over the land and ripped through a whole heap of houses. We were there for about a week and a half, progressively clearing roads with dewar. So essentially, we make a map of every single road, and you start highlighting areas of concern, and you clean it up. The photo on the right here is one of our HEPA filtered vacuum tankers. We own the only ones in the state. So basically a heavy vac that we can, you know, clean up asbestos straight off the ground. You know, lead particles. We've done some firing ranges and things like that. We mobilise straight to site to start working, similar to what we did in Kalbarri. Again, I'm only limited on photographs on this one. Again, examples. This is all ACM material over the road 4 months ago. It's only 4 months ago. Its source was this house here. That was about 350 metres away from it. So it's literally picked the material up and threw it. So this particular street actually didn't have any asbestos structures in the street, but it was impacted, similar to Kalbarri, from other streets and other buildings that had not been remediated. Why do we need to focus on removal? You can see the numbers up on the screen are, you know, a matter of concern still. You know, 6.4 million tonnes, as I mentioned before. You know, we've got a long way to go on this one. And that's it. I'll leave questions to the end.
In the interest of time, we'll take questions at the end and when our next speaker come up. So it's my pleasure to introduce Dr. Melvin Chin. Some fans. So Melvin is a medical oncologist at Sir Charles Gairdner, and he's one of the research leaders here at NCARD. He came to Perth after completing his medical degree at the University of Cambridge in 2011. He has a strong research interest in mesothelioma and explored computational approaches to discover cancer biomarkers based on genes in the mesothelioma. He is a past recipient of the Asbestos Disease Society of Australia PhD scholarship, and he's a current recipient of the RAIN Clinical Research Fellowship. I believe he's going to talk to us about machine learning and AI, and we look forward to your talk.
Thanks, John, for the lovely introduction. And thank you very much for the opportunity to speak today. I recognize many faces in the audience, and these are meaningful faces to me in more than one way. So I appreciate your attendance here. I know John mentioned that it's about machine learning and AI, but the title suggests something different. And I'd like to say that we can reconcile both things. You can have essentially research into more sophisticated techniques, which have a common or unified objective, namely to empower patients to understand what's happening to them with mesothelioma and to help them make choices which are meaningful to them. And therefore, I'd like to focus on the patient-centered component of this, but at the same time, discuss some of the research and updates which have happened in this area since I gave the talk last year. So the main thing that I'd like to start with is that I know in this diverse audience, it's meaningful to me to provide something that is of value to each and every one of you from a talk like this. But of course, because of the diversity of members of the audience, what I've tried to do is to focus on patients, the carers, the advocates, and people who've seen this talk in the past in order to provide some updates, okay? The problem when trying to provide something which is meaningful to everyone is that there are many, many ways in which we can try to understand mesothelioma and to help people affected by mesothelioma. You can do so from the point of view of biomarkers, immunology, novel therapies, clinical trials. So we need to at least condense it into something that resonates with, at least I hope, most of you, okay? So the three themes, and I put them as overarching research themes, are themes which I think resonate with the scientists, with the patients, with the carers. Everybody that comes into my clinic asks me, well, what are the therapies? What are the novel therapies? Everybody that comes into my clinic asks me, well, what are the choices here? How do I decide between the choices? And of course, you could ask this from a patient and carer standpoint, or you could ask about this from a scientist standpoint as well. And of course, everybody that comes into my clinic asks me, as well as that, they want knowledge. They want understanding of why they are making these choices and how to make the most meaningful choices to them. And of course, therefore, I want to emphasize here from this slide that these issues, asking about treatments, personalizing your treatments, asking about why this has occurred and how to navigate these things, this is common. Hopefully, it's a common language that we can all speak. So we'll start first about therapy, and I pitched this, or I explained this in a way which hopefully provides some insight into those who have seen the talk before, or those who have just come here today and have seen the talk for the first time. So welcome. And I'll start by saying that mesothelioma is not a homogeneous disease. It's not just a single type of disease where everybody is treated in the same way, everybody has the same prognosis, everybody has the same side effects of treatment, everybody has the same symptoms from treatment. In the past, traditionally, we used to try to at least provide some guidance or navigation to patients about what this meant for them in terms of outcomes. We divided patients and explained to patients that some mesotheliomas grow quickly, some mesotheliomas grow more slowly, and this, of course, defines a personalized approach to each person's therapy. And of course, we used to say, well, there's the epithelioid subtypes, which on a whole grow a bit slower. There are the non-epithelioid subtypes, which perhaps grow a bit more quickly and perhaps cause symptoms more quickly. And we can see that both under the microscope and also in clinical practice when patients display symptoms. But what we're understanding now is that there's a continuum of things, and the research both here and in other places across the world proves that it is useful to divide treatment into groups, to divide patients in the experience of mesothelioma into groups. But we also need to acknowledge that the continuum between these two groups of patients is actually more subtle and needs to be appreciated when you have a clinical dialogue with the patient about what goes on next. So from a genomic standpoint, from a transcriptional standpoint, from a research standpoint, these categories have fused. And it's actually more important now that for patients, carers, oncologists, clinicians, that they recognize the diversity of phenotypes, the diversity of symptoms in patients, and actually help to personalize that journey for them. There's a second thing which also emphasizes why personalization is important. Now, if I had given this talk to you 8 years ago and essentially summarized what treatments would be, it would be just that single box on the top, chemotherapy. Now there are 5 types of treatments which can be used in the beginning. And essentially, of course, and they're not restricted to being used in the beginning. They can be used at later, even after one line is exhausted or one line stops to work. And this reflects not only the research that has occurred in this area, but the clinical trials across the world. The clinical trials across the world, and also changes in the way in which drugs are accessed. Okay. And the point here is that when a patient steps into the room, or when patients and their carers step into the clinic room, the role of the oncologist now is not just to say, look, here are the side effects, these are your symptoms. The role of the oncologist is to say, look, I need to look at you as a person. I need to look at you for what you are in terms of symptoms. And I need to understand your values in order to provide some sort of treatment which resonates with your values about quality of life, about outcomes, about what you want. Okay. Now, a lot of the research here that's occurring here and across the world is actually about extending that nuance, about extending our ability to provide advice which is personalized. And here, as well as across the world, we study gene expression. So essentially all in tiny cell cancer cells and essentially in the cells around them, what information within those things, within those cancer cells and the cells surrounding them, will help us guide or discuss decisions in a richer, more informative way. Okay. And of course, this is actually coming to patients in Western Australia. It's coming to patients in Australia. There is, although the rollout of these technologies may appear slow, it is happening. There's infrastructure that's being established. All of our mesothelioma patients and cancer patients in general will actually be able to access these technologies that will inform both novel therapies and perhaps better outcomes in the future. And it's close. So what I'm saying right now, just to summarize, well, this aspect about novel therapies or therapies in general, is that we're moving away from saying, look, this is your only option. This is what we need to do for you, into saying that, okay, there are options. We need to understand your perspective about what options you deem best. Because some people might not want chemotherapy. They might have had friends, families, relatives, acquaintances go through it. And chemotherapy is not something that the side effects of those may not be something that they desire. Of course, there are other patients who might say, well, actually, I don't mind that. I've seen those side effects. I think that that's okay. These are things I'm willing to accept. And I want you to look after me so that those side effects are less likely to occur. And of course, we can now have that dialogue because we have choice. So choice is not intimidating. Choice is not worrisome. The fact that you have 5 things that you can use doesn't mean that you should be worried about it. It's in fact actually more liberating than before. So you can have one patient based on clinical genomic information, based on machine learning or AI techniques, might have one treatment. And essentially you might have a second patient where, based on their own personal values about what they feel they want to gain from treatment, as well as the rest of the technologies available to them, will choose a treatment that resonates with their outcomes. Yep. Now let's talk about personalized medicine, because that is something which I think a lot of you would want to know about. And what I will say as a big picture summary is that this is already occurring. Part of the work that's been done here with some of the clinical trials, both the phase 2 as well as the phase 3 clinical trials, have established that we will need to tailor treatment to the individual, and we will need to consider both your viewpoint as well as what information is contained within tumor, as well as in blood information as well. So this concept where you take tumor, symptoms, pathology, genetics, imaging, and time to diagnosis, this is already here. Of course, from the perspective of the patient, we will always make sure that we describe it in a way which resonates with you. However, what I want to reassure the audience of is that when we tell you or when we discuss with you that, look, have a think about this treatment, we're doing so based on an architecture of technology and infrastructure that has been set up across the years. And we're currently at the moment analyzing data from large clinical studies to validate some of the things that we found from the initial studies already. So bigger clinical trials have been done, the signals are there, and essentially the change should take place. So treatment selection is possible. And then we can say, oh, I wasn't able to state that on the slide a year ago, but I can say now that actually you can predict treatment outcomes. I hope to have a conversation in the next few years where we can actually say, look, based on that tumor profile and that blood test, this is what works for you. Is this something that you'd like to have in terms of treatment? And we can also get information as the treatment goes by to say, well, okay, this is something that your treatment appears to be working. It will likely work for a longer period of time than not, based on the information we have. And that degree of personalization is important. You might want to plan a trip to Bali. You might want to spend some time with family. And of course, not in any disrespectful way, but very practical things. You might want to set your affairs in order if you're a patient. And having that information is meaningful. Okay. Now we'll go to the final point, which is knowledge. Okay. And what I want to emphasize here is that, look, there is information, and as patients and their carers and essentially the researchers go through this journey, they are going to be, this is the evolution of what we know about mesothelioma and how we use it is ongoing. So all of you here are part of that story. With everything that you go through, that personal battle that you fight, that is providing information that enriches what we know about the disease. And I will say, as I've said previously, a lot of these technologies, which of course the rollout is slow, but the rollout is there. And essentially, within the next few years, we will see personalized trials. We will send tumors off for sequencing. We are already starting to do that. And in fact, we can do that at scale now. And so these technologies, which generate not only knowledge, this amount of research which generates knowledge, that is being deployed. And I just want patients and carers especially to take away from this that there is choice and there is hope as well. Okay. So in summary, we know now, and I couldn't say this last year, but we know that treatment in mesothelioma should be personalized. The number of drugs that can be used for this have increased. The way in which we approach things based on the research that we know suggests this strongly as well. Okay. And now what we should be working on, which we are doing, both collectively, internationally, is that we're working on strategies to deliver such personalization. Thank you for your time today.
Thank you, Melvin. Again, we'll leave the questions to the end. So, but right now, it gives me great pleasure to introduce Professor Jenette Creaney. She's the director for the National Center for Asbestos-Related Diseases. She heads the Biomarkers and Discovery Unit, investigating improved diagnosis and treatment methods for mesothelioma and lung cancer. So her work includes seminal research on the biomarker mesothelin, which continues to be the gold standard for mesothelioma. And her current research focuses on developing personalized cancer treatments through neoantigen targeting. Without further ado, Professor Creaney.
So thank you all for coming, and hello to anyone online. Yes, so I'm looking more at what we at NCARD are doing. So NCARD is actually about a loose collective of about 30 core members here in Perth, of clinicians, scientists, students, and the lovely Tanya and Thierry, who all work together, but all work on different separate projects. And we have collaborators from across the country and across the world. So, oops, probably should have gone there first. So from a research point of view, we work from the basic biology of what is the tumour cell doing, testing things out in preclinical models, which are primarily animal model systems, and doing various clinical studies and cohort studies. I know many of you are a part of the asbestos review program, and I was just trying to be clever and being with all the cool kids saying computer stuff. But so in terms of us as an organisation, we too have a vision and a mission, but basically our goal is to try and make an impact on the people with asbestos-related diseases, and look to, prevent, treat, ultimately cure asbestos cancer. So today I was just going to talk about some of the programs we've got running across the centre that are primarily looking at new treatment options. So not so much what is currently going on in the clinic, but what we can do in the future. So I thought I might take you back a step to look what is meant by the research journey. So you hear the term from bench to bedside bandied about a lot. And so that basically is just trying to describe the steps it normally takes for research. And it's easiest to describe if you think of a new drug. So the basic science and the biology of the thing is, we get the tumour samples. So we get the tumour samples from the patients who kindly donate them. But looking at the biology of what's going on there and what is going dysregulated there. And then we go, aha, this might work if we fix this pathway up, and test that in preclinical studies. And then after we've tested it in the mice, then we go and test it in the patients. But even the patient testing is a very staged process, and you will have heard about phase 1, phase 2, phase 3 trials. So the first phase is just simply, is this safe? Are we doing more harm than good by giving a new drug to a patient? And it builds up through the phases to, is this actually going to work? And even if we have an effective clinical trial, that still has to meet regulatory approval. So when people say it's 5 to 10 years till we take things from the lab to the clinic, it is a very long process. And I've done this wrong. And I just wanted to point out that it's a process where we start out with lots of ideas and lots of concepts from the early stages, but it all gets whittled down. Things don't work. It's just, it's harmful. And it's also not linear. There's a lot of 2 steps forwards, 1 step back, a lot of circular motion. And unfortunately, it all costs money. And for us, it's the early stage research is mostly funded by the government and by foundations like Cancer Council and RAIN and whatnot. But when it gets to the clinical trials, this becomes a bit more money intensive and frequently requires sponsorship from companies and industry and the big kids. So today I'm going to present you work we've been doing mostly in the backbone of trying to improve on responses to immunotherapy, because immunotherapy is doing something in some people, and we just want it to work in more people and do more. So I'm going to show you some preclinical studies and some of the clinical studies some of you may have been involved in. So the preclinical studies. So I'm going to show you two. One is trying to combine radiotherapy and immunotherapy. As you hopefully know, radiotherapy is just radiation and is targeting the cancer cells and hopefully killing them. But the problem is that it's also damaging the normal natural cells, and that's the terrible side effects. But recently they found out that it's also affecting the immune cells and doing interesting things. And as we know, the immunotherapy is also targeting the immune cells, and it's just an opportunity to combine the two. So we are, as I was saying, this preclinical work, I'm using mice, and basically, very simplistically, give the mice the tumour and then either treat them with immunotherapy or radiotherapy or both. Apparently they won't let us put mice in the CT scanner. I don't know. So we have a magic machine that enables us to actually deliver almost therapeutic levels of radiation to the mice, and that's in the middle picture. That's a tiny little mouse who is being given radiotherapy. So honestly, this is a very simplistic bar chart showing you that in our mouse model, the mice have tumour. If we don't treat them, all the mice die from tumour. If we give them radiotherapy alone, all the mice die from radiotherapy, from tumour, sorry. And if we give them immunotherapy, some of the mice respond. And in this experiment, which when we gave them together, it was like, woohoo, all the mice was cured. But this is, yeah, we would be drinking champagne in the streets. But so this is very specific under experimental conditions. So if we give the radiotherapy and the immunotherapy at exactly the same time, we can cure the tumours. But if we delay the treatment, then there's not such good responses. And sometimes the tumours come back even when so the mice aren't cured, it's just prolonging, which is all good. No, all good. So this is very exciting work, and Naomi has a poster outside if you'd like to talk with her about some of the stuff that's going on. And this project is led by Dr. Alistair Cook, who's not here. And the other preclinical study I wanted to talk to you about was this novel approach that Jonathan here, who's been talking to us, and Kofi Stevens have been working on. Well, looking at combining another type of drug, a drug that's been used in the clinic for donkey's years, to improve the effects of immunotherapy. So this drug is routinely used in this rare genetic condition called Wilson's disease, which the poor people, they have a buildup of copper in their system, and it's not an attractive disease. But one of the curious side effects is you can see here in this picture of their eye, you can see the ring of copper around the iris. So they've got serious levels of copper building up, and their livers in neurodegeneration. But they get treated with these drugs to mop up the copper. And cancer cells, because they're growing crazy like, and need lots of fuel. They actually use lots of copper in the cancer cells, and removing the copper from the system does slow cancer cell growth and also affects immune cells. And I have been told to stress that this is exceptionally preliminary data. But again, a very simplistic bar chart showing you that if the mice have tumours and they're not treated, you can see that they all die in an early or a mid time point. If you give them immunotherapy alone, some of them respond, but over time, the tumours come back. Same with giving the copper drug alone. But if you combine the two, we actually have a more lengthy control of the tumour growth. So, yeah, again, very preliminary work and still very much in the animal model system. But the attractive thing about this is a drug that's been used in the clinic for now 50 years. So the processes, if possible, to get regulatory approval would be shorter than if you had an entirely novel drug. So I was then going to talk about some of the clinical studies. So I was going to talk about 3 clinical studies. So the DREAM trial. So Professor Noack drove the DREAM trial and making the point about how long it takes to get from preclinical to clinical work. Anna, actually, her PhD, and I'm saying it, in 2003, was looking at this concept of combining chemotherapy and immunotherapy. So it's taken 20 years to get to clinical trial stage. So, yep, she spearheaded this trial, and she is also responsible for this acronym that, I'm sorry, doesn't make sense. Durvalumab with chemotherapy as treatment in mesothelioma, the DREAM trial. Okay, there we go. So the phase 2 trial has been completed. And as I was trying to explain about the phases, so one of its goals is to see if it's safe. And yes, the DREAM trial had acceptable toxicities. And the people actually, it was effective in terms of tumours got smaller in more than half of the people. But on a phase 2 trial, you normally only have people who get treated, and you're comparing responses against historical controls. So the whole plan is to do a randomised, multi-centre clinical trial, phase 3 trial, so you can actually compare to see if it is effective in a head-to-head system. So the DREAM, which apparently stands for DREAM phase 3, I don't know what the R stands for, was comparing 2 arms. So the immunotherapy plus chemotherapy compared to chemotherapy alone. So, and people are randomised. So to try and reduce the bias that would be in a trial. Like, this is the test to see if it is effective. And the aim was to recruit about 500 people. Unfortunately, the trial ran across a few problems, not least of which was the global pandemic. But also during this time, as you know, the treatment landscape changed. So rather than chemotherapy being an acceptable comparator for the control group, immunotherapy was being increasingly used. And it just became necessary to close the trial. So this trial has recruited about 200 people, and the plan is to analyse the results early next year. But at the moment, we probably won't see the full results because the whole treatment landscape has changed. So the next trial I'd like to talk about, trial program I'd like to talk about, is the antigen-targeted therapy against cancer. Good acronym, makes sense. Bruce is responsible. So this is a plan to do personalised vaccines based on the DNA mutations of an individual's tumour. So each tumour mutation's different. Yeah. So the plan is to sequence an individual's tumour, identify which mutations are potentially able to generate an immune response, but that response isn't naturally good enough to control the tumour. But if that immune response is boosted, that might be able to control the tumour. So to identify these potential antigens and to design a vaccine, get a vaccine made, and then deliver the vaccine to the patients. So at the moment, we have a phase 1 trial going on. So this is for 6 people. So the aim is to test whether it's safe and it's actually feasible, because it's actually quite complicated to get the tumour, get it analysed, design the vaccine, and get the vaccine, the personalised vaccine made before we can use it. So we've actually did this trial in lung cancer patients to start with, because we used people who we were hoping like we could get the tumour because they were having surgery to cure their early stage lung cancer. So if we gave them the vaccine, it didn't really matter if it didn't work, because these people were potentially cured from their lung cancer. So we've given the vaccine successfully to 4 patients so far. It's all been safe, which was the main aim, and we've managed to do it. So it was feasible. Oh, there we go. No adverse side effects, and we've been able to show that we've been able to make the immune response respond to about 3/4 of the candidates we've put into the vaccine. So for a phase 1 trial, this is looking quite good, and we're still aiming to do the next 2 patients. But moving forward, and probably is we are planning and proposing to do a next trial, a phase 2 trial, in mesothelioma patients. And this will be a collaboration between ourselves and the Peter MacCallum Cancer Institute, Cancer Centre in Melbourne. We're currently applying for funding. As I said, clinical trials can be expensive. And we aim to recruit 36 mesothelioma people. And it's a very, very similar structure to what I just showed for the lung cancer trial, except in that gap there where we're sequencing the tumour, designing the vaccine, making the vaccine. Oops, that wasn't supposed to happen. People are getting their standard immunotherapy, and then they're also getting immunotherapy in combination with the vaccine. And then we'll follow up and see how everything goes. Okay, so the third project I was going to talk about is going back to phase 1 trial, and is, rather than a treatment trial, is an imaging trial in the first stage rather than phase. So this is really quite exciting. This is the Imager trial, and this acronym was designed by our friend ChatGTP. And so at least the letters are the first letter, but you can tell it's doing its normal hyperbole and going evolution and revolution. But this is an imaging trial. So the plan is to compare PET scans using the tracer, the drug that you normally use in the clinic, with a new tracer that is hopefully, well, is called Gerentuximab, G, which targets, for those interested, a molecule called CA9 that is expressed on the surface of mesotheliomas, tumours, and on a range of other tumour types. So topically, this girentuximab has recently been approved for use for imaging in renal cancer, and that's Tuesday's Herald Sun, where they're talking about this recent approval for use in renal cancer, and that this is actually quite an Australian-led project. The company that makes the drug is an Australian company, and we're collaborating with them for our planned phase 1 trial, and they are going to kindly supply the drug for us to test. I don't know. So our Imager trial is to look to see whether this girentuximab does actually see the tumour, light up the tumour, work as good as a PET tracer as our standard current PET tracers. But the really exciting thing is that there's the potential for this drug that binds this CA9, that we can conjugate that to a drug delivery system. So take the drug straight to the tumour rather than giving the drug systemically into your blood, like deliver the drug straight to the tumour. So hopefully moving forward, you'll be able to see in a few years' time that we'll be able to test whether we can use this as a theranostics system is the word we're looking for, which is a combination of therapy and diagnostics. So I've probably gone over time. So I tried to show you before that we were able to do research in from basic biology through to clinical studies, and obviously at the centre of all that is the patient experience. And historically, it's been, let's test things on you. Let's take your tumours and examine them under the microscope. But now we're hopefully trying to move more to making research and the consumer, patient, everyone work together towards this goal of combating asbestos-related diseases. And the plan is to try and figure out what besides can you cure mouth cancer, what needs to be researched, and then help design the research studies, and then also help run the research studies. So moving forward, the whole plan is to make this not just a laboratory, clinician, patient experience, but everyone working together to try and cure cancer. So as I was saying, we've got a big team of people that all work amazingly well together, and we also have support from so many funding bodies and from advocacy groups, and none of it would be possible without the patients and their families. And thank you so much for coming, and please enjoy a beer.
Although we're running a bit over, but we should have some questions and feedback and some discussion going. So if anyone online has questions, please put them in the chat, and then someone here will read your questions when we get to them. But are there any questions for any of our speakers? Just to start off, anyone?
Surely Samuel should have questions gone over.
Is over there.
Hi. In your presentation just then, you talked about treatment naive patients, I think. Is that the term?
Yeah.
What does that actually mean?
So the nursing treatment.
Okay.
So they've not had any treatment before.
Perfect. Thank you.
The virgins.
I was thinking, but yeah, I wasn't.
Sorry, it's politically correct. I'm sorry.
Any other? In the middle of the room.
Forgive me if I missed something, but when you talk about the changing landscape and how the study of Professor Noakes has changed its complexion, and you have 200 people that you're looking at, is that because of parallel investigations that have shown that immunotherapy has changed that landscape, and then you have to reinterpret how the investigation goes forward? Is it because of parallel study has changed the complexion?
So
What you're saying?
so
Should we use the microphone?
no. So because clinical trials are, we have this clinical trial which you like to be enrolled on it. The immunotherapy became part of the PBS scheme. So immunotherapy, which had just been proven to have a 3-month survival benefit over chemotherapy, was now available. It was very hard to recruit to the clinical trial because the immunotherapy is, obviously much more attractive.
Balanced.
Yeah.
Right. Thank you.
The question back there.
Oh, sure.
I had a question for Samuel.
Good.
I really loved your talk. It was really interesting. How do you, because you're contracted by the government, is there not some sort of like legal, not action that you can take, but all these people who are kind of obstructing your work by trying to get onto the site, can you not deal with that as a kind of standpoint as being contracted by the government?
That really depends on the situation, to be honest. Predominantly people are compliant in what we're doing. So something like Kalbarri. Kalbarri actually had police roadblocks. We had it, Bunbury had it as well, but essentially the police would block off road access. They would restrict the area. In the case of Hester, if you're a resident and you were inside and you chose to come out, you actually weren't allowed to return. So the police could put powers of arrest onto you. Obviously, that we don't want that to happen, so we do whatever we can to work with someone wanting to return to their house. There are some situations where we need to clean up something, and we may move our priorities on what we might work with the homeowner and say, look, we need 6 hours, we need 10 hours, whatever it may be. You don't really want to prevent someone of access, but there were certainly situations in Kalbarri where the police were used to remove people because it was a hazard to themselves and to others. Remember that there's not just asbestos hazards. You've got no power, all your food's going off in the fridge. You've got no air conditioning. You've got no essential services. One, a town site is blocked off. There's no, the police are there to do roadblocks, but the ambulance, well, they may not turn up. You've got no mobile phones. If you haven't got a generator, you can't charge your phone, so your phone's going to die. Western Power don't have obligations to maintain certain things as well under emergency provisions. So there's a lot of things that happen that are managed predominantly by the incident control team, and most of the time we're attached into that. One thing that people probably don't realise though is there's something called HEAT, which is the Hazmat Emergency Advisory Panel. So essentially, certain, particularly government agencies, and I've been involved in that a few times, is we'd sit and look at a situation and say, what do we need to do? What do we need to respond to? And media and things like that. So certainly it can happen, but that's an option of last resort.
Would it be right to assume that in your area, the momentum is building after years of struggle?
I feel like that immunotherapy has been a complete game changer. It really has re-energized the system. And I think that's why half of the studies that we do are trying to combine with immunotherapy. Yeah, fingers crossed, fingers crossed.
Yeah, just 2 more questions. Yeah, we can just go. Yeah.
As a participant in the DREAMer trial, how long is it to last, and when will results be?
So the plan is to do the interim analysis 1st January, February, March next year. So, I hope that will be published, April-ish, with a bit of luck. Yeah.
Sylvia, I understand you have a question. So Sylvia, there's a Q&A section. If you can type your question in there, then I'll read it out to the room.
Yeah, I think there was 1 more question. You have... Yes.
First of all, to thank all 3 speakers. I thought excellent presentations. Thank you so much. I wanted to follow up on the vaccine one, which sounds very exciting. Presumably, in a tumour, there are several genes that will have changed. How do you decide which one that you'll develop the vaccine against? I hope this isn't too naive a question.
No. So we're in the current trial, we are using peptide-based things, and we're putting 10 of the top candidates into the vaccine. So using 10 of the mutated genes in the proposed mesothelioma trial, we're going to be using an mRNA-based vaccine. And for that, we can hopefully use up to 20 different of the top candidate genes.
Without being too specific, we weren't concerned. That our major issue with dust, respirable dust coming off the road, was far higher than what was actually on the site. The results were actually surprisingly low compared to what we expected. I'm sure that will be released at some stage in the future.
All right. I know I'm standing in between your beer and yourselves, but let me just finish with a couple of things. So thank you again so much to the 3 speakers. It's really a treat and a pleasure to be able to hear from you. If you have a few minutes, members of the audience, there's a QR code there. You can scan that and just send us your feedback with the public lecture, and any feedback and comments review will be truly appreciated. So the copy of the public lecture will be made available online in a couple of days. So check out.
Give me.
Okay, more than a couple of days. So.
And I'll send everyone who's registered.
Yeah, and so lastly, for our posters outside, just a reminder that there's a popular, popular vote going on. So if you just walk around, find out your favorite, figure out which one's your favorite poster, and there's a box at the table outside where you can put your vote in.
Families. I don't think who's got the biggest family.
It's okay, we'll figure that out. Again, once again, can you just join me in thanking all the speakers, and thank you for